Okay, well, thank you very much for a nice introduction. And first of all, I'd like to thank the organizers for inviting me here to give a talk at what's been voted today as the best university in the world. So it really pains me to say that, but there we are. As mentioned, I'm going to be talking about modeling aspects of solid tumor growth. More precisely, this is what I'm really going to be talking about, because obviously to model something as complicated as this, you can't really put everything in. So we focus on looking at a very small subset of processes with a view to increasing our intuition in a very small way. And the sort of idea is that since there are many groups working on different aspects, that if we can then get enough intuition on different aspects, then maybe we can put all that together and eventually be able to say something. Now, broadly speaking, the research in this area is sort of in two parts. There's a sort of bioinformatics type gene-centric approach, which is to try and look at correlations between presence of disease and genetic disposition to disease. And that's actually quite a powerful way to look at things. And then the other way of doing it, which is the way that I'm going to be talking about, is more a sort of dynamic way of looking at it and basically asking the question that once one has the disease, how do you control the disease? So that's a sort of complementary approach to the first approach. And so I'm going to start off mainly in the talk focusing on this thing here. I'll tell you what these things are as we go through the talk. And these are going to be quite closely related to experiments. And then towards the end of the talk, I'm going to mention some work we've been doing in vascular tumor growth and colorectal cancer, which is more of a theoretical nature, although we are now beginning, literally in the last couple of weeks, to start getting experimental data for this, that we can start to try and link the model to the real biology. Okay, so very, very naively speaking, the body is set up such that it controls cells very well. So cells, if they need to divide, they get a signal to divide. If they're not to divide, they get a signal that basically says don't divide. If there are too many cells, they get a signal that says to kill yourself, and this is called apoptosis, cell suicide. And in this way, the body controls the number of cells that are present. But you can imagine that if these things break down, for example, if you get a mutation so that you no longer respond to the apoptotic signal, or if you start dividing when you shouldn't be dividing, then there's a problem. You get too many cells being produced. And that's basically, in a very naive way, is what cancer is when this steady state, this homeostasis, becomes disrupted because due to mutations, the cells do not respond appropriately to the cues within the system. And what we're asking is, how can we control a growing population of cells? Now, it's been known for quite a long time, since the 1930s, something called the Warburg effect, which is, well, if you think about it, there's two ways a cell can get energy. One is from aerobic respiration, which is using oxygen, and one is from anaerobic respiration or metabolism, which cells undergo when there's a lack of oxygen. Now, this one produces energy, ATP, about 20 times less efficient than normal metabolism. And what's observed is that tumor cells, in many cases, undergo this even when there's enough oxygen available. So there's a question there of why is it worth doing that? Okay, now one argument is that you can actually turn the system around much more quickly if you do this metabolism, so you can sort of turn the crank a bit more and get more energy that way, but it's still less efficient than aerobic metabolism. And in particular, if you think of, look at this as an ecological problem, and you think of cells being in an environment where they're competing against each other, why should you choose to use a metabolism that's an order of magnitude less efficient than the metabolism your competitors are using. Doesn't seem to make sense. Okay? But there's got to be something else to this story. And the acidity of the body, or the body's pH, is set up such that it favors activity of normal cells, as you might expect. It doesn't favor the activity of tumor cells. So when a tumor cell undergoes anaerobic metabolism, there's a byproduct, which is lactic acid, and that reduces the pH of the material, of the surrounding area, to a level that supports more cell division in tumor cells than in normal cells. Moreover, lactic acid is more toxic to normal cells than to tumor cells. So what you can think about it is that the tumor cells are changing the environment to make it more suitable for them than for the normal cells. And so the question is, can that overcompensate for the fact that they're producing energy in a very less efficient way? And this was first addressed by Bob Gatenby, who's a radiologist, cancer radiologist, and Ed Galinsky, who is a physicist, way back in 1996, when they produced this model here. So basically, N1, this is one version of their model. N1 is normal cell density, N2 are tumor cells, L is lactic acid, and everything else, of course, T is time, X is position, and you can write this in 3D. I've just written in 1D. Everything else is a positive parameter. So basically, the idea here is both cell types are undergoing logistic growth. N2, which is the tumor cell type, produces the acid. The acid is degraded. It diffuses. In this version of the model, the acid kills only the normal cells. And here, that lowers the normal cells from their carrying capacity and creates a space for the tumor cells to move. Okay, normal cells don't move. Now, if you look at this, one thing you might immediately say is that, well, if there's space, if this is a space-limiting constraint, shouldn't it be a function of N2 as well as of N1? And one can put that into the model. It doesn't make any difference. Okay? So it turns out when you look at that model is simple enough, and there have been variations of this model, but I'm just going with this model. There's a bifurcation parameter. Just comes out to be one bifurcation parameter. And basically, you get two behaviors depending on the value of that parameter. Okay, now I'm aware that some of you mightn't be able to see this. So this is space. This is level. And what you get is traveling waves. And the waves are moving in this direction. So what you have here is, this is the tumor cell. So basically what's happened, you start off by putting some tumor cells here, and the rest of the environment is normal tissue. And you ask the question, what happens? And the tumor cells are propagating. So here's the density of the tumor cells, coming like this. Here's the density of the normal cells. You can see here a coexistence. And here's a gradient of acid across the front. Okay? And this would propagate in this direction. So finally, you would have a coexistent state of tumor plus normal cells. Now here is, for other values of that bifurcation parameter, here again is the tumor coming like this. And now the normal cells are actually 0 here. So there's been a competitive exclusion, if you like, of the normal cells here compared to the tumor cells. And then, as before, we have the gradient of acid. Another thing to notice is there isn't an overlap here. There's a gap. So this model made by Gatenby and Galinsky, and these are their simulations, make a prediction that you should see a gap between these two populations of cells. So they went back and looked at their experiments to see if they could find this gap. And here you can see in many cases they found such a gap. So here are the tumor cells, here are the normal cells, and here's this gap that's predicted by the mathematical model. And people had done this experiment before to look at the acidity. So here's a tumor, and basically what they did was take slices along different directions and measure the pH. And here the pHs, the interface of tumor and normal cells is about here. This is tumor, this is normal cells, this is pH, which means the acid profile is like this, qualitatively consistent with the model. Okay, and one thing I should mention in terms of the mathematical side of things is that there was a very nice paper last year by Fasano and colleagues where they actually looked in detail at the traveling wave behavior of this system and showed the selection of the wave speed and the existence of traveling waves. Okay, so that is something called the acid-mediated invasion hypothesis. Okay, now life is usually never as simple as that, and so this is work with Natasha Martin, who was a graduate student of mine, and Eamon Gaffney, who's a colleague of mine, and Bob. It turns out that not only is there this competition between normal tissue and tumor cells, there's also cooperation, because not only must the tumor cells kill the normal cells to make space to move, there's other stuff in the tissue as well, extracellular matrix, and that needs to be broken down. And to break that down, you need to use enzymes, which are called proteases, and these are produced in a cooperative way between normal cells and stromal cells, between cancer cells and stromal cells. So what happens if we put that into the model? Now, I'm not going to go into the details of that because the model is very simple. You can easily take the model we've just talked about, extend it to put that cooperation phenomenon in, and if you do that, you get something like this, which is, again, is what you would expect. So here is a measure of the acid production of the tumor cells, and here's the wave speed. So here's the original model where you get this, as you change acid production, you get this wave speed, and you can do the standard analysis to get an equation for this. But now when we put this extension, what we get is something like this. So what's happening here is the tumor is being too aggressive and producing too much acid that's killing all the cells around it, and therefore it can't partake in this cooperative interaction to produce the proteases to get rid of all the extracellular matrix so it can move. Now, the reason why we did this extension was that it has been observed that there are some very acidic tumors that do not invade. So notice immediately what this says about therapy, because when I said that, well, a lot of acid production causes the cells to invade, then immediately if you're thinking in terms of therapy, you would say, let me reduce the amount of acid by giving some buffer. But now you see you could make a problem here. Suppose you were sitting here and you added the buffer, you would make the tumor invade. Whereas if you were here and you added buffer, you would stop the tumor from invading. So you could get the same result. You could get two different results from doing the same treatment, depending on this parameter. Okay. Now this what this also shows here is this is a situation where the tumor, by producing too much acid, has actually encapsulated itself. Now, tumor encapsulation is known to occur. And one theory of explaining tumor encapsulation is that the tumor is obviously a mechanical thing. As it's dividing, it's putting force on the matrix around it. That matrix has got elastic properties, so it can't be infinitely compressible. So it comes a stage where that matrix gets so compressed it doesn't compress anymore. And therefore there's a mechanical barrier to the tumor in terms of its spreading. And in some cases, when you look at encapsulated tumor, that's what you see. You see very high levels of collagen around the tumor, and that seems to be stopping the tumor from expanding. But there are other cases where the matrix around an encapsulated tumor looks perfectly normal. There's nothing wrong with it. And this is not a very good picture for you to look at, but this is precisely what's happening here. This is a tumor expanding, and here is just normal matrix around here. But this tumor is not expanding anymore. Okay, so then this could be an explanation for encapsulation of tumors in this, in this situation. Okay, so I'm now going on to talk about work that we did actually in collaboration with two experimentalists, Bob Gatenby and Bob Gillis, who are now at the Moffitt Cancer Institute, Karen Smallbone, who was a graduate student of ours who's now in Manchester, and David Gavigan, who's a colleague of mine in the Com Lab. And one thing you should be thinking about is that the models I've talked about really could only apply for late stage of tumors, because if you think of tumors beginning with one cell when there's a mutation, then how can I write down an ODE model or a PDE model for one cell? How can I talk about a density? I should really be talking about individual cells. This is what this model does. So this is something called ductal carcinoma in situ. So here is a duct in the breast, and here's a tumor, and here are dead cells. And the question to ask is, why, for example, did a cell get up here? Why should that be a question? It should be a question because cells can only survive if they're in contact with the basement membrane. As soon as they move away from the basement membrane, they die. So how did these cells get to here? And this was a problem that Bob Gatenby and Bob Gillis looked at in this paper here. And they proposed the following situation. They said, let's suppose that these cells undergo a mutation or a phenotypic change, and they become neoplastic, which means they can grow into the domain and still survive. Then you start getting growth to happen, but soon you reach an oxygen diffusion limit, because the only way you get nutrient is from the basement membrane, from the blood vessels at the basement membrane. So there's an oxygen diffusion limit. So now you can only survive if you start doing the glycolytic metabolism. And glycolysis has a diffusion limit that's a bit further away than the oxygen limit. That's not, of course, because this diffuses more than this. It's because this is taken up more than this one. Okay? So now you've had to do two things. You had to become neoplastic so you could move into the domain, and then you had to start using anaerobic metabolism. But you're producing lots of lactic acid, and that's also actually toxic to tumor cells unless you become acid resistant. So this is what's called somatic evolution, an idea that's been around for a long, long time, that the tumor changes the environment and evolves the cell types to finally get a really nasty cell type here, a cell type that can produce acid, kill everything around it, and move, and not be affected by the acid. And then this is the one that usually breaks through the membrane Because see, a tumour in here is no problem. If it stayed here, that's no problem at all. The problem is it breaks through the basement membrane and then goes to other organs. So he asked us to write down a mathematical model of this, and we did the simplest possible mathematical model you could ever think about. We simply took a cellular automaton, so a grid, just a rectangular grid. We stuck cells on the grid, and we said, well, cells can divide, they can die, or they can adapt. So when they divide, they may mutate. And then we said cells use up oxygen, they use up glucose, and they produce acid. So we write down reaction diffusion equations for oxygen, glucose, and acid. And the reaction parts of those reaction diffusion equations come about from doing some biochemistry. So we write down equations like this for production of energy, and then we simply use law of mass action to convert those into equations. Okay, and then we give rules. For example, you look at a cell, you look at what reactions are going on at that cell position, you then figure out how much energy does it have. If it's got not enough energy, it'll die. If it's got a lot of energy, it can then divide. And when it can divide, there's a certain probability that it will mutate. So lack of ATP, it'll die. Proliferate if there's enough ATP above a certain threshold. And then we have we read off the acid concentrations, and if the acid concentrations are very high, the cell will die, but that probability is scaled by the cell type. So if the cell is acid resistant, it'll have less chance of dying than if it's not acid resistant. And then we get this sort of picture here, which you'd immediately expect. This is a cut into the duct, and you see here, this is the basement membrane, this is oxygen, and then this is glucose, because here oxygen is very low, glucose is being used up, and the glucose produces acid, the acid diffuses, so that's all what you would expect to get. You don't need to do a mathematical model to come up with that. Run the simulations. Because it's probabilistic, you can get different ways to go. So here's an example where we go from being normal, hyperplastic, glycolytic, and hyperplastic, and then the really nasty types. But sometimes when you run the simulation, you go this way. You go from there, to there, to there, and to here. So what's happening here in this transient is you're getting large nodules of glycolytic cells and small nodules of glycolytic cells. So little islands of glycolytic cells. And I must confess and say that we didn't really think anything about that, but Bob, the experimentalist, said, well, this is a prediction of the model. I'm going to go and do the experiment and see if I can see these blobs. So off he went and did an experiment, and these red things are blobs. The staining stains red for cells that have upregulated the glycolytic pathway. And so what you see here is like big blobs and little blobs, nodules all over the place. So very much qualitatively in agreement with the model here. Okay, and then that's further details in this. So now if you think of therapy, what therapy should you do? An obvious therapy is add bicarbonate to neutralize the acid. Okay? And so you add bicarbonate. So this is a poor mouse, had a tumor implanted here. You add bicarbonate basically by giving bicarbonate in drinking water all the time. And this is to show that even though the body is great at buffering, it does change the bicarbonate. It does change the pH. And now the question you ask is that if if the cells are using acid to invade, then when you add bicarbonate, you should see less invasion of this tumor in the mouse that is drinking bicarbonate. Okay? So now this, I apologize for the quality of this. These are now looking in the lungs. So basically, you add the tumors there, and then you kill the poor mouse, and you look at the lungs. And this is untreated, and the fluorescence, you mark the cells with fluorescent dye, and where you see the fluorescence here, those are tumors. So there's a lot of invasion. Here's a treated mouse, and you can see very little. And this is better done here. These are the number of invasions with an untreated mouse versus the number of invasions of a treated mouse. So by adding the acid, by adding the buffer, which neutralizes the acid, you are actually, at least in these experiments, slowing down the ability of the tumor to invade. Okay. Of course, it's not very important to cure cancer in mice. It's very easy to cure cancer in mice. The problem is what happens if you go to humans. So what we've done recently is write down a mathematical model to try and compare mice with humans. I'm not going into the detail of it. So basically we consider, talk about coarse graining, we consider the body to be composed of blood and tumor. Can't get more coarse than that, okay? And we basically follow acid. And so basically acid can arise from the production from the body, through ventilation, from production from the tumor, and can be affected by renal filtration. And so you write down a mathematical model for that. It's a compartmental model, so again, very, very simple to write down. I'm not going to get into all the details of writing it down. But what we do is that we know that some of these parameters are different in humans than they are in mice. So this is the rest of the equations. Again, these are all very standard, and actually, to be honest, pretty boring. And these pictures here, the take-home message of these pictures here is that because you have different parameters in humans than in mice, when you look at the effect of buffering in humans, the model predicts it's going to have a lot less effect than in mice. Okay? So we, being mathematicians, said, oh, that's no problem at all. Just give lots and lots more bicarbonate. To which the response was, well, bicarbonate is toxic, so you'll poison the patient. Okay, so we said, oh. Okay. So what we did then, it turns out this model has 3 different time scales. So from the point of view of a PhD thesis, this is fantastic because you can do lots of asymptotics. From the point of view of being something useful, it also means that you now have analytic expressions for everything, and therefore you can do a parameter sensitivity analysis in an analytic way. And then you can ask the question, what other parameters are these results sensitive to? Because if we can only manipulate the bicarbonate in a certain way because we want to kill the person, are there other things we can manipulate with the human? And our analysis, so we go back and we do all this. I'm not going into the details again, actually can't really see these things. These parameters here, red parameters here, are the ones to which the model is most sensitive. And so that has suggested that we do a combination therapy. In fact, it turns out it's most sensitive to proton production. And this thing here, DCA, is a proton inhibitor. So we've made a suggestion that one should do a mixture of bicarbonate treatment and proton inhibitor treatment. And this is starting to be done by colleague Ian Roby in Arizona, starting to do these experiments on mice to begin with. Do combination therapy and see whether this is better than doing the simple therapies. Okay, our model also predicts things like, for example, looking at the kidney functions, drugs that lower this parameter, which are things like aspirin, ibuprofen, etc., would also help, and that some blood pressure drugs could also help as well. But of course, all that needs to be tested experimentally. Okay, so that's viewing really the results of the first part of the talk. Acid mediates invasion, somehow get rid of the acid, and you can stop the tumor from progressing. Now, it's known, more and more studies are coming out, that exercise can delay the onset of cancer, particularly breast cancer. And that seems to contradict this modeling, because what happens when you exercise? Well, one thing that happens is you get cramp. Why have you got cramp? It's because of lactic acid. Where did that lactic acid come from? It's because your cells have run out of oxygen, they've undergone the glycolytic pathway, and they're producing lactic acid. So basically, when you go for a run or do some exercise, you're dosing yourself with acid. Okay, lactic acid, not the drug acid. Okay, because I gave a talk once in a place which was a bit rough, and they thought I was talking about hallucinogenic drugs. But I wasn't. Okay, so what happens now if we take the second model we talked about with the somatic evolution and we pulse it with acid? So every day we give it half an hour of acid. Okay, from a mathematical point of view, very, very simple thing to do. Okay, what we end up finding, again with Karen and Bob, is that you change the evolution, the somatic evolution, and you select, at least in the model, a different phenotype. You select a phenotype that's hyperplastic. So this is the nasty one, hyperplastic, glycolytic, and acid resistant. What our simulations show when you start hitting the thing with acid is you select the phenotype that's hyperplastic and acid resistant. And this phenotype is not invasive. So the suggestion there is that what you're doing is you're modifying the environment to select a less nasty phenotype. So really two approaches. One approach is change the environment to neutralize what the tumor is using in order to invade, namely the acid. The other approach is to change the environment to select for a different cell type that's less invasive than the previous cell type. And what we're hoping to do is actually get a graduate student to start working on this, and then we're wanting to do these experiments on the poor mice again in the lab in the Moffitt with Bob to try and test this hypothesis. Okay. So now I want to leave that and just for the last few minutes talk about, and just talk this very briefly. This is very much work that's in progress in the sense that we don't really have a tie-up with experiments. And in fact, we started all this off about 10 years ago, and we thought, you know, what would be really good, what would be really, really nice would be if we could have a model of the vasculature of a tumor, because we want to figure out how to get drugs into a tumor. And then if we could make predictions of how that vasculature changed over time, and then we could work with an experimentalist. Experimentalists could give us an image, a nice image of a vasculature. We would put that into our model, and then we would predict that in 5 days' time the vasculature would look like this. And then the experimentalist would image 5 days' time, see what the vasculature looked like, and we could do a comparison. Okay? We thought, oh, fantastic, this would be really great. Okay? So that was 10 years ago, right? the first experiments were done 2 weeks ago, taken 10 years, because of course, again, being mathematicians, we just think this is a very easy thing to do. So the experiments just started 2 weeks ago, so I can't really comment on experimental results, but I'll tell you about the model anyway. Okay, and this is work with you people will know Helen Byrne from Nottingham, Marcus Owen from Nottingham, Tomas Alacon, who was a postdoc of ours, of Helen and mine, and he's now just moving to Barcelona, and a couple of summer students, James Murphy and Russell Bettridge. And basically, this is a cellular automaton model again, but the difference between this one and the last model is we have a vasculature. And we have dynamics going on inside the cells. So basically, we have a vasculature. It brings oxygen into the system. The cells use up the oxygen. When the oxygen is very low, due to little dynamics that we have going on inside the cells using ordinary differential equations. Of course, we use reaction diffusion equations to describe all this stuff. These cells produce VEGF. VEGF diffuses into the surrounding medium. It does two things. It causes these blood vessels to widen, and it causes the process of angiogenesis. That's the process by which cells in the blood, on the blood vessel break up and start to migrate towards the tumor and form more blood vessels. Okay, so we have this coupling across the scales from tissue scale to what's going on inside the cell through a mixture of ODEs, ordinary differential equations, going on in here, discrete model for the cells, and PDEs for these things here. Okay, the vasculature we model as an adaptive structure. Oh, here's what we do. We do it in 2D to begin with, but we have some 3D later on. There's a very nice series of papers by Tim Seacombe and Axel Priest, so this mathematician and experimentalist, on vascular adaptation. And the question they ask is that if I know the radius at time t of a blood vessel, what's its radius at time t plus delta t? And the answer is it's the radius at time t plus some stimulus. And what's that stimulus? The stimulus is composed of a mechanical stimulus, which is due to wall shear stress. Basically, the blood vessels try to adapt so that they have a certain wall shear stress experienced. Metabolic demand related to what the surrounding tissue wants. And when they put these in and they analyzed rat mesentery, which is where they were looking, they found that this would not give you the right structures that they were observing. So they postulated two other, there should be shrinkage. So basically, if there's no stimulus, the vessels will shrink and disappear. What they found, they needed to have the short range and long range stimuli in as well. And when they did this, they were able to produce results that were consistent with what they observed in the experiments of normal vasculature. Okay? Now, one thing to point out, this is for rat mesentery, and rat mesentery does not get cancer. So what does this say about normal cancerous tissue? Well, we don't know, but we're going to take this as the model. Okay? So basically what we do, we set up the system, we put flow through, using Poisson flow, through the vasculature. We change the radius depending on this thing here. Then that determines the amount of oxygen going through the system. That determines how the cells divide, how they produce VEGF, etc., etc. That affects back on this, changes it, and that you feed back and forth. Okay? And typically the picture you get is this one. So again, this is blood vessels. These are normal cells, and here's a tumor. The tumor starts to grow and then starts to run out of oxygen, around here. These cells start to produce VEGF. The VEGF causes more oxygen to come in, and the thing continues to grow. That's basically what you get. Okay? So while we were waiting for the experimentalists to start giving us images so we could do something with this, we decided to play around with this model. Now, it's known that tumor vasculature is not normal. It doesn't respond in the normal way that normal vasculature does. So, of course, we can play games. We can say, well, if we manipulated these different responses, what would the tumor look like? And then we could look at real tumors, and from that, try to do the inverse problem and figure out what could be going wrong with the vasculature. So, for example, if we modify this thing and find that the predicted sort of characteristics of the tumor doesn't look like any tumor, then we could say, well, that's not the way things are happening. So we did that. And we concluded, by looking at gross pictures like this, that losing these long-range stimuli look to be what makes tumor vasculature different from normal vasculature. And in fact, more recently, Tim Seacombe actually did a really nice job of this, where he actually looked at the vascular structure. And because now these vascular structures are available, so he did a 3D simulations of what his model would predict of what the vasculature would look like, and then by looking at them was able to say, well, that looks like a tumor vasculature, that doesn't look like a tumor vasculature, et cetera, and came up with the same result that we'd come up with the 2D model. Okay, I'm not going to go into all this detail. I'm just going to go to this picture. So in terms of therapy, you could give, there's something called anti-angiogenesis treatment. The idea is very simple. If the tumor is producing angiogenesis factors that is causing the vasculature to go to it, if you can stop that from happening, the tumor will die. So you can produce anti-angiogenesis agents. Apply that, and you can stop angiogenesis from occurring. And when that was put forward, actually quite a long time ago now, it was thought that this was the cure for cancer, and unfortunately hasn't worked out that way. Okay. Now, another thing about cancer is that, as I mentioned, cells divide when they shouldn't divide. Therefore, if you could develop a drug that targeted dividing cells, then you could kill cancer cells and not kill normal cells. And that's what stage-dependent chemotherapy does. So now you might think, why don't we apply both? Okay. And then if you think again, if I apply anti-angiogenesis, I stop nutrient getting to the cells will stop dividing because they don't have any nutrient, and therefore the chemotherapy will be totally ineffective. Right? Seems to make sense. In real life, if you do this, you find there's a synergistic effect. Chemotherapy and anti-angiogenesis, by and large, works better than one or the other. So that seems to contradict our little verbal reasoning model. Okay? And here's one possible explanation for that. Don't look at this, just look up here. What we did here, this is total tumor cells depending on different densities of vasculature. Here, a high density of vasculature, and each one of these has the same amount of oxygen in them. So here, high density of vasculature, lower density of vasculature, very low density of vasculature. And what you can see happening here is as we lower the vascular density, we increase the tumor load. The reason for that being that we've actually done a vessel pruning that's made the system more efficient at taking taking oxygen into the system. And this is what the body does naturally. The body sets up your vasculature system by laying down the system and then pruning it so that it gives a very efficient way of delivering nutrients. And according to our model, this is a situation where this is what anti-angiogenesis is doing. Now you hit this with chemotherapy and you wallop it. So you have the synergistic effect. Okay. So recently we've gone to 3D, and this is with Holger, and I'll try and show this video now, but it was taking quite a long time when I was showing it earlier on. So we'll see what happens. So what you should see if this comes up is a block of pink cells, those things there which are normal cells, 2 blood vessels, here and here. Now these blood vessels are not here, they're actually in here, right? But this is for visualization, we put them out here. What we're going to do is put into this a green vessel, a green cell, which will be a tumor cell. And then the tumor cell will start to grow. When it runs out of oxygen, it'll start to produce angiogenesis factors. That'll start to cause these blood vessels to dilate and start to grow. And what we'll end up with will be a complete mess of, so if we go here to half size, so we run that, so just you can see all sorts of things like that happen. Okay. End up with a very nasty looking thing. What does that mean? Nothing until we get experimental results that we can link this model to. And Mark Lloyd at the Moffitt Cancer Institute has just started doing these experiments a couple of weeks ago and is now trying to measure things like branching angles, things like frequency of branching, lengths of branches, numbers of tumor cells in implanted tumors in mice. And then we're hoping to try and take this data and try to parameterize this model and see if it can be of any use. Okay, but at the minute, its use is just to produce rather nasty looking pictures. Okay. So the last thing I want to do, and I just want to mention this, oh, that was a big mistake. Is to mention something that's a bit mathematical, and this is John here, sitting there. We've started to model the crypt, and I don't want to go into details of this because I don't have time to go into details, but this is modeling colorectal cancer. And this takes mechanics into account, because those of you who love mechanics are probably getting ready to kill me because I've spoken for 50 minutes on a biological topic and not mentioned mechanics once. So I apologize for that. This takes mechanics into account in a simplistic way, but it's beginning to take them into way. But one of the things, and we end up producing pictures like this, I don't want to go into the details of these sorts of pictures where we can sort of basically look at how growth occurs, etc. These are based on taking cells and assuming that they're linked by springs. And then you basically solve Newton's laws for each one of these centers. That moves the cells around, and then you ask the question about cell division, etc., etc. As I say, I don't want to go into the details of that, but I want to show this from the point of view of there are many different ways of doing cell modeling. I've focused on cellular automaton models. Here's another way of doing it by looking at how the center of cells move. Another way is vertex models, which is looking at solving Newton's laws for the vertices And another way of doing it is to write down a Hamiltonian, a potential, and say that cells move down the potential gradient. And there may be other ways, I don't know. The question asked then is, which is the correct model? And the answer is, it's the model your supervisor used. Okay, and that's not a very scientific answer. So what would be nice would be, could you in some way write down a model, a mean field type of model for each one of these? So one thing you could do is you could say, let's do millions and millions of simulations and compare these models. And that's very difficult to do because the parameters, there's not a one-to-one correspondence between parameters. So what to do? So could you write down a mean field approximation to these models and then look at those and compare them to each other? And now we've done this in a very, very simple case of this type of model, actually using ideas from polymers. And what we find out that we get is, and this is with Philip Murray, who's a postdoc of mine, and Karina Edwards, who was a postdoc, and Marcus Tyndall, who's now at Reading, is with a very, very simple model, we get something like this, where Q is the cell number density, and these are parameters. So what we have here is a nonlinear diffusion, and that's simply arising out of these cells moving around because of the elasticity against them. Okay, and what you can see here, for example, is this model doesn't make sense if Q goes to 0. And that corresponds to the cells being very, very far apart, which actually wouldn't be in real life in this system. Now, in the meantime, people like Mark Alber up in Notre Dame in the US has been using sort of statistical mechanics type approach to look at the model of moving down the gradient, potential gradient. And they come up with an equation like this with a different nonlinearity here. And what you can observe is that for quite a big range of this value of Q, both models agree. But in the limits, they don't agree. And those are limits where, in one limit, our model doesn't make sense, and the other limit, their model doesn't make sense. So now maybe this is a way to start to distinguish between these models. Having said that, this is for the simplest possible setup, and as soon as you make a thing more complicated, we don't know how to do it. So see that as a challenge for the future in terms of then really trying to understand mathematically what's going on with these models. Okay, and then this is when you do millions and millions of simulations and try to compare the models, and it's not so easy for the reason that I mentioned. Okay, so I'll finish there. What are the key take-home messages to sort of say that we used very, very simplistic models to begin with, ODE models, individual-based models, to look at cancers in different ways, to look at it as almost like an infection, a pest in an ecological setting, and ask the question, if in an ecological setting you had a pest, how would you deal with it? And the answer is, well, in ecology you do not deal with it by trying to eradicate it. We know that doesn't work in ecology. In ecology you develop ways of manipulating the environment to either make the surrounding population stronger or make the environment worse for the pest or drive the pest to a different phenotype so it's not so resistant. You basically try and make it a refuge. And what we've tried to do here is essentially do that, but in the context of ODEs and PDEs and cellular automaton models, and suggest various therapies, some of which are now presently undergoing some actually in people as well as in mice. And then the last bit of the talk was really trying to get across the idea that when you get these multi-scale models, and you need multi-scale models if, for example, you want to understand the combination effects of chemotherapy and anti-angiogenesis, both of which act on very, very different scales, so you need a multi-scale model to understand those. The multi-scale nature of the problem can give you counterintuitive results. And finally, that you really need to develop some sort of mathematical theory, or we really need to develop some sort of mathematical theory that really stops us from just doing computations all the time and tries for us to be able to compare these different models with all the different parameters they have, but from a mathematical context rather than from a brute force numerical approach. And I should just mention here that there are many, many other models addressing very, very many different aspects of this problem, but many with the same sort of philosophy in mind. It's just using different processes. So I'll end there and thank you for your attention.